This lecture is on amino acid metabolism. We look into amino acid, which has shown on the structure there, the carbon skeleton, which composed of the carboxylic portion, hydrogen, as well as the R side chain, as referred as carbon skeleton. This will be removed from the amino group, and the carbon skeleton will be utilized in the energy production from the amino acid, while the amino group, being toxic at high levels in the cell, required to be converted to urea in mammal by the urea cycle. The source of amino acid metabolism used for catabolic pathway is actually from two sources, that is from dietary protein, protein from meat, which will be digested and eventually will form free amino acid. The other source is actually through the in vivo production of amino acid, that is through transcription and translation to produce free amino acid. Another thing from the diet as well as produced amino acid within the body is also you have to look into those that has been used to make tissues such as muscle cell. So all these can be utilized, however need to be regulated. If too much has been taken from tissues such as muscle, can lead to muscle wasting, and that will be quite detrimental to the body itself. So here we begin in terms of understanding the catabolism of free amino acid. Firstly, the amino group need to be removed by the effect of this reaction referred as transamination, as done by aminotransferase. This aminotransferase has the coenzyme pyridoxal phosphate (PLP), which assists within the active site of aminotransferase in its reaction. So, as seen in the third paragraph, firstly, the amino group will be reacting with the alpha ketoglutarate to form glutamate. So, you will have the amino group taken out from the amino acid, leaving behind carbon skeleton, and the amino group will become glutamate. Then, in the second reaction, which is referred as oxidative deamination done by glutamate dehydrogenase, the amino group will be now free again and reform alpha ketoglutarate. So this is the two steps where the free ammonium is obtained or attained from the amino acid. This slide actually repeats what's been described in the earlier slide. So here the amino group transferred to the alpha ketoglutarate to form glutamate, and then another reaction to yield free ammonium ion. This also describes the aminotransferase, where the enzyme relevant to transamination, the first step itself. And this also been mentioned here, the aminotransferase could be also used in the anabolic pathway to obtain amino acid. You will see later the aminotransferase been described in terms of the function of PLP in transamination reaction. So here a bit more on that first reaction, where the alpha ketoglutarate, which happens to be the mitochondria Krebs cycle intermediate, will react with the any particular amino acid. There are 20 amino acids, and therefore will then produce the glutamate and also the keto acid. This keto acid, the carbon skeleton, will then proceed for metabolic in any metabolism, such as the keto acid could be oxaloacetate, could be keto pyruvate, and utilized in the energy production. So this is the oxidative deamination reaction, the second step, whereby the glutamate will then become something of an intermediate shift base, and then followed by releasing the amino group to become free ammonia and regenerate alpha ketoglutarate again. So after the free ammonia is obtained, it is quite toxic for the cell. The more and more buildup of free ammonia can cause toxic effects to the cell, and therefore it needs to be removed or detoxified. In terms of detoxification, you need to see which organism that it is found. If it is terrestrial vertebrates, then it is converted to urea. Therefore, urea cycle is of importance. If it is water organism found in water like fish, it will be just secreted as ammonia. So we have to look into which organism this ammonia is released upon. And another important thing about this slide is the release of the free ammonia after the two reaction is actually located in the mitochondria, which happen to be useful because mitochondria is also the beginning of the urea cycle. So it facilitates in the detoxification of the free ammonia. So this slide gives you a summary of that two reaction, that is transamination followed by oxidative deamination, where it gives rise to eventually free ammonia and as well as the carbon skeleton, which compose of alpha keto acids such as pyruvate. In terms of the free ammonia, excess will be made into urea, while some also will be utilized in the biosynthesis of nitrogen containing intermediates or end products. In terms of the figure given, that is on the diet obtained amino acid, as well as those found made into tissues such as muscles, you have to realize that urea cycle only found in liver cell, the hepatocyte. When it comes to utilizing amino acid from diet, as well of that found connected to muscle, then some form of adjustment required to be done when it comes to the excess of ammonium ion. If the ammonium ion is not transported to liver, then it will stay in these tissues and will cause toxic effects. So in order to transport this ammonium ion that is being generated, the body has two transporting amino acid, that is alanine as well as glutamine, which we will look into in the next slide. So as been referred at the end of the explanation of the previous slide, there are amino acid found in other tissues such as muscle, such as kidney, that need to be also utilized for the amino acid. For that purpose, two principal transport form of amino acid, just for the purpose of transporting the excess of ammonium generated to be brought to the liver. One is by alanine, while the other is by glutamine. So here been referred further where the alanine cycle as well as glutamine are useful. Uh, Better you see the next slide where shown in detail about the transportation from other tissues other than liver in terms of being brought as alanine as well as glutamine. Alanine, once it arrives from, say, from muscle, it will be broken down to pyruvate in the hepatocyte, and then the free ammonium will be then also released. When it comes to glutamine, which comes from also from other tissues, muscle and also brain, therefore it is also brought to liver, where it is to release the ammonium ion, and the ammonium ion all will be then made into urea by urea cycle. Shown also in terms of the what happens in the liver, where cellular protein, where through transcription, translation, also from diet, is actually found in the liver, and that free amino acid, due to two successive reaction, will then bring out the keto acid, the carbon skeleton of the amino acid, as well as the free ammonium, where need to be detoxified using urea cycle. Next, we look at the urea cycle, which will be described in the few slides to come. The urea is made up of one contributing by the free ammonium, and then also the other amino group contributed by the amino acid aspartate, while the carbon given by the bicarbonate ion. So that is the structure of urea as shown in the slide before this. When it comes to the ureothelic organism, such as mammal, human included, they dispose the free ammonium ion after being converted to urea, while other organism been described in the next slide. When it comes to the urea cycle itself, this is one of the three cyclic pathways that you have learned. The other two cyclic pathways are the Krebs cycle as well as the Calvin cycle. Interestingly, this cycle, the urea cycle, has also been discovered by the same person, that is Hans Krebs. So here summarize in terms of how the excess of ammonium is excreted out into the environment so that it is not toxic to the body of the organism. In terms of how it is excreted, there are three forms. One, urea, that is for the ureothelic animals, terrestrial vertebrates, such as rat or even humans. Then you have the uricothelic animals, such as birds. Birds are migratory animals, therefore they need to go from one area to next area a long distance. Therefore, they tend to use energy not only from carbohydrate, also up to the level of using protein, amino acid. And therefore, since a lot of use of amino acid for energy, there will be also a lot of release of ammonium ion. And therefore, they need to find a way to remove excess of ammonium ion before it becomes toxic. And the form that is useful for that purpose is actually in the form of uric acid, where it can dispose for double the amount of nitrogen compared to just being urea. Then you have also the aquatic animals such as fish, where they do not have a reason to convert to become urea or uric acid, because as soon as ammonia is produced, it can be immediately disposed from their body into liquid environment surrounding them, and therefore immediately it can be diffused away from their body, and that is the water environment. So there's disposal is just by ammonia itself. Here shown the symptoms of ammonia intoxication, as well as what causes the ammonia intoxication. In terms of the cause, it is definitely the failure in the important organ relevant for urea cycle, that is liver. So if anything to go wrong to liver, therefore excess of ammonia could build up in the body. And then also in terms of the urea cycle itself, perhaps one or two enzymes, crucial enzyme in the urea cycle being defective, it could also lead to accumulation of ammonia, and that could be intoxicating. The symptoms of intoxicating can start from mild up to the level of being really toxic to the body, that is coma, and also death could come about. The symptoms include anorexia, that is a person who losing weight, uncontrolled loss of weight, and further on you have other symptoms also been shown here. This slide is just a repeat of what's already been described to you. It's just referring back that excess of ammonia is highly toxic, and it has to be brought to the liver to be detoxified by converting to urea in the urea cycle, and also other tissues such as brain, intestine, muscle also need to utilize free amino acid. However, the free ammonium ion that is released will be brought to liver by the transport form of ammonia, that is glutamine and alanine, to be detoxified. So a little bit of the biochemistry related to the urea cycle itself. The first three step is in making this intermediate referred as carbonyl phosphate, and all this happen in the mitochondria of the liver, where the free ammonia will be reacting with the bicarbonate to form the carbonyl phosphate with the assistance of ATP. And these three steps takes part in one single enzyme, that is carbonyl phosphate synthetase, which happen to have three active sites. So this is the three steps being described taking place in the carbonyl phosphate synthetase. The first step in the first active site is to activate the bicarbonate to become carboxyphosphate, and then the free ammonium ion comes, and there will be a switch from the phosphate to become carbomic acid, and then comes the next ATP hydrolysis, which eventually will make into carbonyl phosphate. Here, just to take into the most important part of the text, so the carbonyl group that is found in the carbonyl phosphate will next react with ornithine to form citrulline. Here shown the reaction itself, where carbonyl phosphate will react with ornithine to give rise to citrulline, which become much larger molecule itself. Both this ornithine and citrulline are in fact qualified to be amino acid. However, they are not part of the 20 amino acids since they do not form any structural proteins. Again, here I just want to highlight the important tags whereby the reaction so far that we have been described about urea cycle is taking place in the mitochondrial matrix. The next three steps, that is from citrulline, will then come out from mitochondria and go into the cytoplasma to carry out the next three steps. So therefore, it can be concluded that this urea cycle compared to Kelvin cycle as well as Krebs cycle, this one involves two different locations. So here once citrulline is in the cytoplasma, it will then react with amino acid of aspartate to form arginosuccinate. This is followed by arginosuccinate breaking down into arginine as well as fumarate. And then due to the arginase found in the liver, it will cause the arginine to become ornithine as well as finally urea itself. Here given the urea cycle summary, and this diagram I think I have utilized it for final exam already twice, and it has been used in assessing the students. You can check the final exam question on how I have asked question for final exam. But basically the urea cycle described in this diagram shown it happens in the mitochondria as well as the cytoplasma, and the end result, it prevents hyperammonia. Just to briefly describe it, you have the bicarbonate ion, the carbon part of it is reacted with the free ammonia to become carbonyl phosphate by one single enzyme having three active sites that cause three steps to be done to produce at the end carbonyl phosphate. The carbonyl phosphate will react with ornithine to form citrulline. Citrulline goes from mitochondria to cytoplasma to react with aspartate to build something which is much larger, arginosuccinate, and this will then break into fumarate and arginine, and eventually arginine breaks even further to become urea as well as ornithine. Ornithine goes back and repeats this cycle. So here once the urea is the end product, it will be then secreted mostly in the kidney, that means as urine, as well as small amount, that is 5% in the feces. I also have provided URL links on the YouTube videos that gives you further description of this urea cycle. Now we look at the fate of the carbon skeleton, that is after the removal of the nitrogen part of the amino acid. So once the amino group is removed, the amino acid is left with the carbon skeleton, which could be either pyruvate, acetyl coenzyme A, acetoacetate, as well as certain intermediates of citric acid cycle. Which one of these will be formed is all dependent on the 20 amino acid that loses the amino group. Therefore, the 20 amino acid can be actually classified as to be glucogenic if it is to give rise to pyruvate as well as the intermediates of citric acid cycle, and it will be referred as ketogenic if it ends up to be acetyl coenzyme A or acetoacetate. There are also certain amino acids that can be both of these ketogenic as glucogenic. The meaning of ketogenic and glucogenic is referring to what can be made into ketone bodies, hence ketogenic, or could be made into glucose through gluconeogenesis. So here given the table to separate the 20 amino acid into glucogenic, ketogenic, as well as to be combined glucogenic as well as ketogenic. They are separated to these three categories or classification based on whether they end up as citric acid cycle intermediates such as alpha ketoglutarate, oxaloacetate, or pyruvate, and classified as ketogenic if they form acetyl coenzyme A, acetoacetyl CoA, or acetoacetate. So this figure is better to describe the classification of glucogenic type of amino acid, which indicated in the red boxes, while that of the ketogenic type of amino acid based on the yellow boxes. So you have, for instance, alanine will form pyruvate. Instead of going through the citric acid cycle, it will then enter the glucogenic pathway by becoming oxaloacetate and then further steps until become glucose. You also have, for instance, the isoleucine forming succinyl-CoA and then can enter to become glucose through gluconeogenic pathway. Here, the yellow boxes indicate, for example, isoleucine forming acetyl-CoA, but instead of going to become citrate, it's more, encouraged to become acetyl-CoA and therefore enter the ketone body production rather than becoming citrate. And then you also have, in terms of certain amino acid, to be forming both glucogenic as well as ketogenic. For example, the phenylalanine amino acid can either form acetyl-CoA or form fumarate. Therefore, it can be classified as both ketogenic as well as glucogenic. So here some form of a summary of the usage of amino acid coming either from intracellular protein that is built within the cell through transcription, translation, or perhaps even derived from tissues such as muscle cells, and also in terms of diet protein being digested until to become free amino acid. So all these amino acids firstly need to be removed out the nitrogen part of it to become ammonium ion, as well as the keto acid. The keto acid such as pyruvate, alpha ketoglutarate, oxaloacetate will enter the citric acid cycle or even utilized in the formation of glucose through gluconeogenesis. So from amino acid, you can get glucose as well as getting energy from it. In terms of the fate of the ammonium ion, not all is actually toxic. Some are in fact utilized for the biosynthesis, for instance, the base structure of the nucleotide. But the excess cannot remain in the body because it is toxic. Therefore, it is converted to urea before it can be excreted out of the body. We now continue in terms of understanding the anabolic pathway relevant to amino acid metabolism. We're going to look into how the amino acid will be made within the body itself by utilizing amino group coming from glutamate as well as glutamine to be connected to keto acid, which derive from three pathways, that is pentose phosphate pathway, glycolytic pathway, as well as Krebs cycle. So in supplying the amino group in making new amino acid, the amino group can be uh, sourced outside of the organism body itself. That is the environment, the nitrogen that is found in the atmosphere. This nitrogen can only be made into nitrate or nitrate, which cannot be done by the animals because it requires the breaking of the three bonds found in nitrogen. However, the nitrogenase that is found in the legume nodules of the legumes, leguminaceae plants, they are able to break this nitrogen to become nitrate, and that is where the source comes from. To understand the source, we have to look at the nitrogen cycle, where the atmospheric nitrogen content up to 78% to 79% can be then, due to the microbes that is found in the nodule of leguminaceae plants, then can what you call make it into ammonia, nitrate, and then nitrate, and that through can be assimilated into plant, and then by plant to the animal, and then eventually to the humans itself. So here I have another diagram on nitrogen cycle. I prefer to use this diagram for nitrogen cycle, whereby you can see that atmospheric nitrogen can be made into, can be through acid rain, it could cause to form nitric, then nitrate, due to lightning that causes this breakdown of the nitrogen bond. But that's not going to be plenty. Uh, the one also possible is through the effect of the plant, leguminaceae plants, that can absorb this nitrogen and then cause it to become nitrate and then nitrate. And then when the cattle eats the plant, and then you consuming the cattle, there is been a shift in terms of the nitrogen from atmospheric to that into the body of the person itself. So once the nitrogen coming into the body as ammonium ions, not only from, from the atmosphere, the ammonium ion could already pre-existing in the body itself. So assuming the free ammonium is now within the body, it needs to be made into this two amino acid, that is glutamate, and then followed by glutamine. These two, glutamate and glutamine, is considered the donor, major nitrogen donor for whatever biosynthesis that requires the amino group. Uh, for instance, from glutamine, it can be made into tryptophan and then histidine amino acid. So it's supplying the amino group and react with the keto acid. Therefore, finally, you get the amino acid. This slide, as well as next slide, uh, describe how this reaction of taking the free ammonia and connecting it to alpha ketoglutarate to finally obtain glutamate. Here is shown the mechanism of how the glutamate is made from this ammonium ion, and this one involves shift base. I'm not going to go in detail about it, but if you look at the next slide, this is just to describe the general formation of shift base, while here you get to see the actual reaction that comes about in making glutamate, that is alpha ketoglutarate reacting with ammonium ions, forming this shift base intermediate, and then finally forming glutamate. At the above of the slide here, it doesn't stop at making glutamate alone. After that, glutamate eventually can be made into glutamine with the intermediate shown there. So in a summary, based on what is found in the atmosphere, the nitrogen, eventually it become ammonium ion, and this ammonium ion can, through a series of reaction, can be made into glutamate as well as glutamine. So now you have the donor of the amino group within glutamate and glutamine. You now need the carbon skeleton. The carbon skeleton is provided by uh, intermediates of glycolytic pathway, pentose phosphate pathway, as well as citric acid cycle. In terms of synthesizing the 20 amino acid, it's not a problem for plant and microbe. Plant and microbe can synthesize all 20 without any issues, because they have the pathways, the enzyme concerning in making all the 20 amino acid. Unfortunately, over or through evolution, human beings cannot make all 20 and only can make 9, has been indicated in this slide. Those that cannot be made within the body and required to be obtained from the diet are classified as essential amino acid. And apart from essential, non-essential, there's also something referred as conditional amino acid. Conditional amino acid, one example is arginine. In humans, arginine cannot be or insufficient when in the form of babies or growing child. Uh, but when already grow up, arginine is sufficiently provided by the urea cycle, and therefore the amount is sufficient. So conditionally, when in young child, it's not sufficient, therefore it's considered like essential. Then when grown up already, it's sufficient, become non-essential, therefore it's considered called as conditionally conditioned amino acid. So here given description as I just explained in the previous slide. So you have the listing of the non-essential, essential, as well as conditionally essential. So already spoken to you about the arginine, that is conditionally essential, differ in terms of youth as well as adult. Then you also have in terms of essential, number of amino acid shown there, uh, and also non-essential. And then I also told you microbes and plants, no problem. However, humans, they need to obtain the essential amino acid from diet. Then there's also the asterisk there on tyrosine. You see, the some amino acid need to be made from essential type of amino acid. For instance, tyrosine is made from phenylalanine. So if phenylalanine is not provided sufficiently in the diet, Therefore, it cannot be made into tyrosine, and this means tyrosine becoming now essential. So it all dependent on the amount of phenylalanine that can be obtained from diet to decide whether tyrosine is essential or non-essential. Here, a bit of story on how, in terms of essential and non-essential, the non-essential appears to be synthesized in the body by using very few steps. The pathway requires very few steps, while the essential type ones, studying in terms of plant and microbe, has given indication it requires a number of steps much more than that of non-essential, 5 to 16 steps itself. This can be seen here in this, the figure there, where you can see the essential ones will require at one amino acid even up to 16 steps, while the one non-essential up to the most is 4 steps, and there are some 5 or 10. But generally speaking, the non-essential one require very few steps compared to the essential type of amino acid to be synthesized. So what can be concluded is, over evolution, humans, or rather mammals, have lost the ability to synthesize this essential amino acid within the body itself, because the pathway is no longer present. The crucial enzyme is no longer present in the body, simply because perhaps the body start to realize that uh, takes a lot of effort to make that amino acid, but easily can be obtained from the diet. So why bother making it de novo itself? Rather, just get it from the diet, especially mammals being omnivores. So next we're going to look into this reaction, one particular reaction in detail, that is transamination by aminotransferase. We're going to look into the mechanism of this aminotransferase, where it takes the amino group from glutamate and then link it with the keto acid, which could be either alpha ketoglutarate, oxaloacetate, or pyruvate, and make it into amino acid. So we're going to look into transamination in terms of mechanism of the enzyme aminotransferase. So this is another one of those slides that's going to talk about the mechanism of the enzyme. It's quite complex, but as been told to you that I'm not going to assess on the mechanism of any particular enzyme, as well as regulation of any particular pathway. So just for your understanding, this is on transamination reaction related to aminotransferase, and it has aminotransferase has PLP, pyridoxal phosphate, which serves as the coenzyme. So this is the description of the mechanism of the enzyme itself, that is aminotransferase, that's performing the transamination. I will use the highlighter here. So you have the lysine amino acid connected to the protein part, which is not been shown there, together with the PLP. This is the PLP. So the PLP, pyridoxal phosphate, is the crucial part of the active site that takes part in the reaction itself. Then the glutamate comes, it provides the amino group. It itself will be converted to alpha ketoglutarate. So here you have the amino group already connected to the PLP. Then comes the what you call keto acid to actually provide the carbon skeleton. So now both the carbon skeleton as well as the amino group will be connected to the PLP, and then without going in detail further, a few of the mechanism of the enzyme take place in order to finally cause the release of the L amino acid, and therefore that explains the mechanism itself, where crucially PLP plays a major role in the mechanism. This slide is basically to describe the state of the PLP in two forms. That is the two tautomeric forms. Not much to describe further on it. So in the synthesis of 20 amino acid, you need to have a donor of amino group. Then you also have to have the keto acid. The keto acid can be provided by the intermediates of glycolytic pathway, the citric acid cycle, as well as the pentose phosphate pathway. Can't talk about all 20, how each one of them being synthesized, but as an example, here given the synthesis of serine as well as glycine. It starts off with the phosphoglycerate, which happen to be the intermediate of glycolytic pathway, and then it will be converted to 3-phosphohydroxy pyruvate. So this is the keto acid. Then comes the donor of amino group, that is glutamate, and it will be changed into phosphoserine. And then once the phosphoserine is removed from the phosphate group, then you get serine, and then from serine you then can get glycine. So now we look at how the 20 amino acid is synthesized in terms of the origin of their keto acid. So you have in blue pentose phosphate intermediates providing the keto acid, and then you have in red from glycolytic pathway providing the keto acid. So from there you can see various amino acid can be synthesized. While here shown the sources of Krebs cycle or citric acid cycle that actually provides the keto acid in making the other types of amino acid. While here provided the summary of all the amino acid being synthesized from the precursor of the keto acid, just to highlight the fact that although here been shown 20 amino acid, but you have to realize that not all 20 is being made in mammal organism. Plant and microbe only can make all these 20, while mammals only can make around 9 amino acids.